Well, hello, welcome to today's lecture by Dr. Esther Guzmán. We're going to fix the sound a little bit. You got it? Thanks. That's Wyatt. Wyatt is our IT person who really makes it very helpful so that we get this to you every week. I'm Dennis Hanasak, the research professor here at Harbor Branch and host of the series. And we look forward to resuming lectures to our audience here at Harbor Branch after the pandemic subsides. But for now, we're connecting with you virtually. So in 2 weeks, on Wednesday, February 10th, we have what I like to call a 2-for-1. 2 speakers, 1 topic. And our speakers will be Dr. Dan McCarthy from Jacksonville University and Dr. Karen Holloway Atkins from East Coast Biologist, Inc. Their lecture is titled A Look into East Florida's Nearshore Hard-Bottom Reefs. And Dan and Karen are part of a collaborative team that recently published the first book ever about Florida's nearshore hard-bottom reefs, which is a highly productive habitat that needs greater recognition. And that book, which I have right here with me, I actually read it over the holidays. So this is what scientists do for holiday breaks. We read books like this. It's a really good book. It's called Islands in the Sand, Ecology and Management of Nearshore Hard-Bottom Reefs of East Florida. And I've already told my colleagues that scientifically, I think this book is a terrific contribution on these important reefs that are literally just a few steps from our beaches, extremely shallow. We're not talking about deep things. And the lecture really will be a tour along our coast here. And you'll see some of the 90 color pictures that they had in the book that bring these reefs and its plants and its animals to life. So along the way, I bet that we all learn a lot of things that we did not know before. I certainly learned a whole bunch of things. And again, it's right on our doorsteps to the ocean. So I really hope you can attend our 2-for-1. Now, as always, it's a pleasure to introduce today's speaker, Dr. Esther Guzmán. She received her Ph.D. in Immunology from the University of Texas MD Anderson Cancer Center in Houston in 2004. And then she had postdoc positions at the Henry Ford Hospital in Detroit, and then here at Harbor Branch. And in 2007, Esther became the lead of our cancer cell biology program in our Center for Marine Biomedical and Biotechnology Research. And this program focuses on drug discovery for cancer. So Esther and I were just talking, it's been more than 15 years already. I remember when she first came, and her work over that time now has resulted in more than 30 publications on that topic, and also 3 US patents. So today, and by the way, Esther only talks every 3 years. It's kind of on a schedule. So, you got to wait another 3 years to hear this. But today she's going to give us an update on her work in her lecture, The Deep Sea's Treasure Trove, Marine Natural Products as Potential Cancer Therapeutics. Esther, it's great to see you.
Great to see you too, Dennis, and thank you for that kind introduction. And with that, let me just get you started. Okay, so I'm going to talk to you today about natural products and marine natural products. So one of the questions that we always get is why explore the seas for medicines? And so marine natural products are also known as secondary metabolites. They are small molecules, and it requires energy to make them. So we believe that even though they're not essential for survival because they require energy, they must confer some evolutionary advantage to the organism that makes it. So we also know that many marine organisms produce these marine natural products to better compete for resources and survive. And as many of you know, the oceans cover 70% of the Earth, and they hold about 50 to 80% of the planet's biodiversity. And this biological diversity translates to chemical diversity. So when you have a beautiful reef like we do here, that has so many different organisms present, you can imagine that it has that many chemicals present as well. So marine natural products or natural products in general, not just marine, but actually mainly terrestrial, are the basis for about 62% of the medicines that we currently use. The best example of this is aspirin. which I carry in my purse. And aspirin comes from the bark of a tree, but it is now produced in the lab, and they change it slightly so that it's stable carrying it in my purse. But it is a natural product that we use very commonly. And over 48% of cancer medicines are also traced back to natural products. So the beauty of natural products is that they are bioactive. Mother Nature is a great chemist. The reason they get made, we might not know, but they have some sort of biological activity. And that makes it that they have the potential to be used for our health. So, and indeed, there are already marine natural products that have been approved as cancer treatments. So we have, for example, jaundice or trabectedin or actinium 743. This is derived from a tourniquet. Scientists here at Harbor Branch Oceanographic Institute at least figure out its chemical structure. This compound has been approved by the European Commission to treat soft tissue sarcoma and ovarian cancer, and it was finally approved in 2015 by the FDA to treat soft tissue sarcoma. Another example is halaven or eribulin, which is a synthetic analog of the marine sponge natural product halicondrin B. And this was approved in 2008 to treat late-stage metastatic breast cancer for patients that had already undergone 2 different regimens of chemotherapy. In Europe, it is the first line of treatment for metastatic breast cancer, and hopefully it will evolve here in the US to also be that. But you can see that these compounds are very useful. So it is not easy to translate our compounds from my lab to the clinic. It is a process that takes a lot of time. And so here I'm giving you the example of actinium 743. It was first extracted in 1969 when they found to have anti-tumor activity at NCI. It took until 1990 for its chemical structure to be figured out. And as I said, Dr. Amy Wright here at Harbor Branch had something to do with that. So go Amy. It took until 2000 before PharmaMar found a way to make it in the lab in a manner that was commercially viable. And then in 2007, it was approved by the European Medicines Agency and for the use to treat soft tissue sarcoma. And it took until 2015, as I told you before, for the FDA to approve it for the same reason. So it's a long process, but I hope to convince you through my talk that these compounds have some amazing activity that makes them worth the investment of time and money. So Harbor Branch Oceanographic Institute has had drug discovery here since 1984 under different names. Many of you know us as the Biomedical Marine Research. Our official name now is Marine Biomedical and Biotechnology Research. But our mission is to discover marine natural products that are useful against dreaded diseases. And this is a multidisciplinary approach. We have many groups. We have a natural products chemistry group that is led by Dr. Amy Wright, who also leads the whole division. And this is, it isolates and purifies the natural compounds. They do the chemical characterization of compounds, they figure out their structures, and they have worldwide collaboration to test against many diseases. We have a microbiology group that is led by Dr. Peter McCarthy. And this does isolation and culture of marine microorganisms. They also test for activity, antibiotic activity and antifungal activities. And they also do environmental assessments. My group is the cancer cell biology group. So we do screening to find activities against cancer. And we also try to determine how the compounds do those activities. We have a sponge cell biology group that is led by Dr. Shirley Pomponi, and they're trying to create cell lines from sponges to produce the compounds. They're also trying to figure out why the sponges make those compounds, how it benefits them. And finally, we have a biosynthesis group that is led by Dr. Woyun Wang, and it identifies the genes that are responsible for producing the compounds. And they can also manipulate the genes in the lab to produce more compounds or similar compounds while they're producing more. So you can think of it as they produce compounds, we test them for activity, and this is, and also Peter, here at Harbor Branch. And both Charlie and Woyun try to figure out how to obtain more of them so that we don't have to go back to nature. So before we get really into the nitty-gritty of it, I had to introduce you to two key concepts in cell biology. I know I'm going to toss these words at you, and so I would like to define them before we move forward. One of them is apoptosis, and the other one is signal transduction. So apoptosis stands for programmed cell death. And it is a normal process by which a cell that has finished its function, it can eliminate itself, or if the cell has been damaged or has become old, this is the way that a cell will get destroyed. It takes energy, it's a very orchestrated process. And in cancer, this process can be disrupted to allow cells that should have died to continue living. Many cancers are resistant to apoptosis. And this is a problem because a lot of chemotherapies work through apoptosis in order to exert their function. So one example of apoptosis that I think everybody has heard about or has encountered is when you have an infection, your white blood cells, which include T cells, B cells, and many others, they gather and they start multiplying in order to destroy the infectious agent. Once that is done, you get rid of the excess white blood cells through apoptosis because their function is finished and we don't need to have that many. Now, the other concept that I want to introduce to you is signal transduction. Wikipedia defines signal transduction as a process by which a cell converts one kind of signal or stimuli into another. And cells use signaling cascades to divide, to produce growth factors, to produce new blood vessels, to undergo apoptosis. So this is the way that cells operate. And the best explanation of signal transduction came from Amy, and I stole this from her because I think you will agree that this makes perfect sense. So if I were to order a pizza and things go well, the delivery guy gets into his car and he follows the traffic signals, or he or she. And if everything goes well, the outcome is we get pizza. Everybody's happy. However, if they get into the car and they encounter a signal that is not functioning properly, what's going to happen is you get an accident and there's no yummy pizza. So to translate that now to cancer biology, if you think of a normal cell, this is a cell where all the traffic lights work. So all the signaling pathways work. This cell will only divide when it needs to divide. It can undergo apoptosis if it's necessary, and it will only grow in the tissue of origin. A cancer cell where the signaling pathways are defective, proliferation signals are always on, so this is a cell that is constantly dividing. It's not able to undergo apoptosis, and it is able to grow in site different from its origin. So this is a way to understand a little bit of what signal transduction is all about. So I work on cancer, and I always like to start my talks by defining that cancer is not a single disease. It is the general name for a group of over 100 diseases. What they have in common is that you have cells in part of the body that are growing out of control or failing to die, and these cells contain damaged DNA. Now, that damaged DNA can be caused by inheritance. It can be the result of exposure to chemicals, viral infections, smoking, excessive sun exposure, to name a few. But this is to tell you that there's not going to be one cure for all cancers. So some summary statistics to kind of get you thinking about this disease or these diseases. Cancer affects 18 million people worldwide with over 9.5 million deaths annually. 1 in 3 Americans, and some say it is closer to being 1 in 2, will have some form of cancer during their lifetime. And sadly, cancer risk increases with age. So we're living longer, which is the good news. But the bad news is that we're seeing more cases of cancer than we used to see in previous decades. Cancer is the second leading cause of death in the United States. It is preceded only by heart disease. And in 2020, we had an estimated 1.8 million new cancer cases with about 600,000 deaths. However, I know that a lot of people feel like we have been researching for cancer for so long, how come we haven't done better? And the answer is we are doing better. We are making a lot of progress in our fight against cancer. It's just that it is not over yet. So deaths from cancer in the United States have decreased about 25% since 1991. And this drop is due mainly to reducing smoking, and also advances in early detection and treatment. And these advances in early detection and treatment are the result of the investment in research. The more we know the disease, the more we can find ways of either prevent it or actually treat it. And so that has led us to come to the age of what we call personalized or targeted medicine. Nowadays, if a patient goes and has a biopsy of a potential tumor, we have means to look at the receptors or the molecules that could be residing at the surface of the cells. And based on that, know what therapies might be effective against that. We also can do genetic sequencing, and that allows us to know what mutations the tumor might have, and also what signaling pathways might not be working properly. So all this knowledge put together might allow us to devise a treatment plan that is very geared to that particular patient with that particular tumor and help us solve cancer that way. So I'm going to tell you a little bit about cancer drug discovery here at Harbor Branch. And so many of you know that for many years, we were very lucky to have access to a ship and a submersible. Now we have access to remotely operated vehicles that allow through the Cooperative Institute. And this allows us to collect marine invertebrates. These marine invertebrates are then extracted with solvent, fractionated or separated into different compounds. And this leads to the creation of what we call the PIC library, which are enriched fractions that contain a mixture of 3 or 4 compounds. And of course, we have a pure compound library. These libraries are then sent to screening. In-house, I do the screening for cancer, but they are sent to many other labs in the US and throughout the world to test for different activities. Once an activity is found, we work with the chemist towards the purification because they purify the compound, trying to go for the compound that is responsible for the activity. We call that bioassay, guided fractionation. And then they go along and figure out the structure. And for us, after we obtain a lead, we try to figure out how is it working. So we do what we call mechanism of action studies. So some of the sample collections here at Harbor Branch, as I said, we had access to many tools that allow us to collect many marine organisms. From those marine organisms, we have a collection of about 19,000 marine microorganisms and about 30,000 marine macroorganisms that can be used in our drug discovery efforts. I told you about the organisms then get extracted. And the easy way to think about an extract is if you make tea or if you make coffee, you're making an extract. I subsist in coffee, so I use that as the example. And so you can see you have the coffee beans, you ground them, you combine that with hot water, and you get this wonderful drink that contains caffeine, which is the chemical that we all think about, but it actually has 1,000 other chemicals, which is what gives it its flavor and its smell. And it's a similar process with tea, but whenever you're doing that, you're making an extract. Our chemists use a similar approach. They grind the organism. They use different solvents, so they don't use hot water. They tend to use alcohols or other solvents to make it liquid. But from that, they obtain different fractions that are then purified and put either into the peak library when they contain mixtures about 3 or 4 compounds, or if it's the final compound, it goes into a pure compound library. So the reason we like to use the peak library is that, for example, I'm showing you here a sponge, and this sponge has many compounds that kill cancer cells, but it has other compounds in addition to that. And by having these enriched fractions, we can focus on the activity and actually find those other compounds. So you can see here that by following the green activity, those boxes where we have the green, we obtain this compound that has actually IL-8 inhibition. And IL-8 is a cytokine, it's a soluble factor that is produced by cells, but it facilitates the production of new blood vessels. So if a tumor is making interleukin-8, it's gonna generate new blood vessels and it's gonna grow. We don't want that, and this compound will prevent that from happening. If you focus on the yellow activity, you end up with this completely different compound, and this has CCL2 inhibition. So CCL2 is another small soluble factor that is produced by cancer cells, but it has a totally different function. It serves to attract immune cells to the site of the tumor. And those immune cells, while they can fight the tumor, some of them produce growth factors that allow the tumor to grow. So by preventing those immune cells from ever reaching the tumor, you are also preventing those growth factors to get there, and the tumor cannot use them to grow. The other tool that we use a lot in my lab is what we call cancer cell lines. And so whenever a patient goes, has a tumor and has surgery to have that tumor excised, if they get permission, the tumor is dissociated into single cell suspensions and is trying to grow in the lab. This is not a trivial process. It takes a lot of effort. You can ask Dr. Pomponi about how difficult it is to create cell lines. But if it grows, then it is placed in a repository, such as ATCC, where scientists like me can order these cell lines. And the beauty of these cell lines is that they keep on dividing and they keep on growing in the lab, and they retain the characteristics of the tumor that originated. So if the tumor was a metastatic tumor, that cell line retains that metastatic potential. If the tumor had signaling molecules that were not working, that cell line will carry those. If the tumor arose from particular mutations, that cell line will have those. So it makes it very easy for scientists like me to study my compounds on humans without hurting anybody. So we like to use those. So the type of testing that we do, we have two main kinds. We call it cytotoxicity screening, one of them, which is shown here in the purple. And this simply tests the ability of the compounds to kill cancer cells. Of course, it's not as simple as that. We want what we call selectivity. We want to kill many cancer cells for each normal cell that we kill. And if you think in terms of patients that are undergoing chemotherapy, many of them are losing their hair or throwing up because you're killing normal cells at the same time that you're killing cancer cells. We're trying to get away from that. And so we're trying to look for compounds that have that selectivity that kill many cancer cells for each normal cell that they kill. The other kind of screening that we do is called target-based phenotypic screening. And it's huge names. And this scary image is the way that we think of signal transduction. So here you can you see like a molecule that activates many other molecules that serve to activate different pathways. But what we do in this kind of screening is we focus on one of them and we see if we can change it. If it's always on, can we turn it off? If it's always, if we have a lot of it, can we bring it down? Can we bring those levels down? And so we call that approach forward chemical genetics. We know we're changing a phenotype, but we don't know exactly where we're acting. So if I were to bring down this molecule, I don't know if I'm acting right here or much here. So I need to do what I call mechanism of action studies to try to figure out where exactly in that pathway I'm making that change. So as I said, I'm talking to you about the drug discovery program here at Harbor Branch, but I'm kind of giving you the cancer cell biology perspective. And I'm going to focus specifically in 3 areas of investigation that we're following in my lab. So these include activating the immune system to eliminate tumors, selectively target signaling processes that are activated in cancer, and then using more tumor-like 3D cultures. For all of this, I use a technique called high-content imaging. This machine that you see here is a high-content imager, and it's basically an automated microscope that rapidly images samples and analyzes for selected responses. This is the overall approach that we follow. We plate our cells and we allow them to adhere overnight. The next day, we replace the media with media that contains our marine compounds or controls. We normally incubate for a set amount of time, which is usually 24 hours. We fix, and then we label with an antibody for our target, a cell mass that defines the body of the cell, and a nuclear stain that shows where the nucleus is. And we obtain results that look like this. So here's an example of cells treated with solvent control. And the red in there that you will see is normally our molecule that we're targeting. And this is an example of one treated with our natural product. And you can see that there's a lot less red. So the beauty of the high content imager is that it quantifies it. It doesn't only rely on what my eyes are seeing, but it gives me a certain percentage of how much I brought it down, which for me is insanely useful. The other thing is that it allows us to sample a lot of samples at a time, or to test a lot of samples at a time. So this is an example. If you have a test tube, you can run one test. In the previous assays that we used to do, we used 96 well plates. So every single circle that you see here would be the equivalent of that test tube. And so we were able to test 48 samples in duplicate at a time. Now we're using 384 well plates. And so every single one of these wells represents one of those test tubes. And now with this setup, we're able to test 192 samples in duplicate at a time. So it has allowed us to have much higher throughput. So the first focus on my story is going to be on pancreatic cancer. And pancreatic cancer is the 4th leading cause of cancer death in the United States. You can see in 2020, we had about 57,000 new cases diagnosed, and we had about 47,000 deaths due to this disease. But I want to call your attention to this graph. So the light green shows the incidence or the rate of new cases. And sadly, this is moving in the wrong direction. You can see that it is increasing. And this is not what we want to see with any kind of cancer. And the lethality rate or the death rate, which is in the darker green, is also slightly increasing. So again, this is not where we want to be in our fight with cancer. And to make things even sadder, you can see here the 5-year relative survival is 10%. And that means that patients that were diagnosed, only 10% of patients diagnosed with pancreatic cancer survive 5 years post-diagnosis. So I hope that that graph and that number show you that we have to find better treatments for this disease. And if that didn't do it, this scary graph will. So there was a report that was put out in recent years that has shown that unless we change the way we are diagnosing and treating this disease, pancreatic cancer will become the 2nd leading cause of cancer death in the US by 2030. So why is pancreatic cancer so aggressive and so difficult to treat? Well, the main problem, one of the main problems that we have for this cancer is that there are no early detection methods that currently exist. Now there's talks of approval of a blood test that will help to detect cancer, so pancreatic might be included in that. And if that happens, that should improve our statistics. But it's not only that, those tumors are highly metastatic. So that means that they spread to other organs. And normally when patients are diagnosed, the cancer has already spread to other organs. The tumors are resistant to apoptosis and to current chemotherapies. There are many risk factors for pancreatic cancer, but one of them is pancreatitis or inflammation of the pancreas, which increases the risk of developing pancreatic cancer. And pancreatic cancer also has many unusual ways in which they grow. They can activate unusual pathways to obtain nutrients. And they can also use their microenvironment, what cells they bring to be surrounding them to produce growth factors that they use to grow. So the way that we are approaching, one of the ways that we are approaching pancreatic cancer is by a method called cancer immunotherapy. And I'm an immunologist by training. And so all of those immunologists that work on cancer will tell you that the easiest, most elegant way to eliminate cancer would be to get your immune system to recognize that tumor. So why that doesn't happen? Well, it does happen. There's a theory that says that the immune system is constantly screening and finding tumors and eliminating them. But cancer has gotten smart and has found ways to evade being recognized. And so in order for a T cell to recognize a cancer cell, that cancer cell has to have particular receptors or changes the receptors that it expresses to avoid that recognition. So we're trying to work on those receptors to try to see if we can make our own immune system recognize the cancer and fight it. So I told you, so there's a cytotoxic T cell or killer T cell that if it sees a cancer cell and there's the presence of the right receptors, what's going to happen is that that killer T cell will kill the cancer cell and you will get apoptosis. However, if the cancer cell expresses a molecule called an immune checkpoint, such as B7H4, what you get is a weaker response. And so that is something like this.
We don't need to see his identification.
These aren't the droids you're looking for.
These aren't the droids we're looking for.
He can go about his business.
You can go about your business.
So, and to put it in more current event context, it's not Bernie that you're looking for, but basically an immune checkpoint is similar to in Star Wars, Obi-Wan Kenobi using the Force to tell the guards to ignore them and let them be. By having B7H4, the cancer cell is telling the T cell, just let me be, don't do anything to me. Obviously, we don't want to be with Obi-Wan Kenobi in this one. And so the way we are trying to approach this is to use a small molecule to either bring B7H4 down or prevent its binding. And once we block that immune checkpoint molecule, we should restore the response and get apoptosis back. And indeed, this is successful. So there, I don't know if you guys have seen this, but there's drugs against different immune checkpoint molecules that are currently in the market that are being used in the clinic successfully. And these include PD-L1, PD-1, and CTLA-4. But sadly, as you can see by the list of cancers in which they are active, none of them is pancreatic. None of these immune checkpoint inhibitors works in pancreatic cancer. But pancreatic cancer does express very high levels of B7H4. So here you have pancreatic cancer cells, and you can see them in the blue, that's the nucleus. There's a little bit of green in some of them, which is the body. But all the red that you're seeing in this photo is B7H4 expression, and this is just our control. If, however, you treat with one of our compounds, one of our pure compounds, hopefully you will agree with me that we brought B7H4 down. And this should make these cells more susceptible to being recognized by your immune system and be able to undergo apoptosis. The other way that we're focusing on immunotherapy is by targeting a molecule called FOXP3. And so I told you antigen presentation relies on having the right receptors, but there are other molecules called co-stimulatory molecules, such as CD28 and B7, 1, and 2. And if these are present at the time that antigen presentation occurs, you actually get a much stronger response. So while when you're fighting cancer, you want this stronger response to occur. If it happens at other times, it leads to autoimmune disease, and you don't want that. So your immune system has a whole way in which it can prevent very strong responses from occurring. And this is by using its own T cells called regulatory T cells, which are recognized by different markers they express, including FOXP3. And these T cell will then come, and if there's a too strong of a response being presented, it would actually bring it down. But a new finding has shown that some cancer cells, such as pancreatic cancer and colon cancers, express FOXP3. We don't know why they do it, but we believe they're using FOXP3 as the Obi-Wan Kenobi, convincing the guard to let the cell be. And so again, our approach is using a marine natural product to try to bring those levels of FOXP3 down or to block its binding to restore a response. And so again, these are pancreatic cancer cells, but now the red that you see is FOXP3. And hopefully you can see that when we use this peak library fraction, we brought the levels of FOXP3 down. And so the cool thing about this is, again, it should make it susceptible to being recognized by the immune system and to undergo apoptosis, but we still don't understand what FOXP3, how it works in the cancer cell. So a molecule like this could help us study that and understand it even better. So to summarize this area of the talk on cancer immunotherapy, we have identified marine natural products that target B7H4, which is an immune checkpoint. And these compounds have the potential to be therapeutics for pancreatic, colon, breast, and lung cancers. We have also identified marine natural compounds that downregulate the molecule FOXP3. And these have the potential to be therapeutics for pancreatic cancer and colon cancers, and also to understand the role of FOXP3 in the disease. And we are in the process of writing grants to support this research to continue. So fingers crossed, send me good vibes so that we can continue this research. So the next part of my story is going to focus on colon cancer and lung cancer. And so I will start with giving you the statistics. Colon cancer, we had about 150,000 cases last year with about 53,000 deaths. Now the incidence and lethality for these are trending the right way. They're going down. We want to see that. And patients are surviving. About 65% of the patients are surviving 5 years post-diagnosis. So we still see a lot of deaths. We still have to target it, but we're seeing better numbers with this particular cancer. Lung cancer is the main killer, is the top leading cause of cancer death in the US. You can see that we had about 230,000 cases last year with about 135,000 deaths due to this disease here in the US. Again, both the incidence and lethality are trending the right way, but the 5-year survival is only 21%. So only 21% of patients survive 5 years post-diagnosis. So again, we still need to do more work towards lung cancer. And so the way we're tackling these two cancers is by focusing on signaling molecules that are not working properly in cancer. And the molecule that we chose to focus on is called survivin. And survivin is involved in the regulation of cell division and the regulation of apoptosis. It prevents apoptosis from occurring. So you can think if you have high survivin expression, this cancer cell has enhanced cell division, it cannot undergo apoptosis, and it's also resistant to chemotherapy. If we use a marine natural product to bring down the survivin levels, now we have a cancer cell with lower survivin expression that is they can undergo apoptosis and cannot divide anymore. So this is the whole approach that we're focusing on. And so here's the example. So now the picture here, these are lung cancer cells, the A549 cell line, and the red that you see is survivin. And hopefully you can see the effects of the compound. So these are a peak library fraction that reduce the levels of survivin about 76%. And it comes from this lovely marine organism. This other peak library fraction caused an 86% reduction, and it comes from this marine sponge. So to give you a summary of this area, we just concluded a 3-year grant from the Florida Department of Health, the Bankhead Collie program, that allows us to identify inhibitors of survivin in both colon and lung cancer. We identified this novel activity of bringing down survivin levels in cancer cells for 6 non-marine compounds. And our chemists did a fantastic job and isolated through bioassay guided fractionation 20 compounds with this activity, of which 10 are new. And sometimes you hear people saying that we cannot find any more new marine compounds. So here's Amy proving them wrong. And these compounds are potential therapeutics for colon, breast, and lung cancers. So the last part of the story that I'm going to talk about focuses on breast cancer. Breast cancer is the second leading cause of death for US women. There were about 275,000 cases last year and about 42,000 deaths. And again, here the trends are kind of steady. We are not increasing, but we haven't decreased the rates of new cases. We are decreasing the rate of lethality, but not as much as we like. And even though the 90% of the patients survive past year, 5 years past diagnosis, many of them have recurrent forms of the disease, and there are still forms of breast cancer that are very lethal. One of those is triple negative breast cancer. And triple negative received their name because they lack estrogen and progesterone receptors, and they do not overexpress the epidermal growth factor receptor, or HER2. So there are no receptors that can be targeted to treat this particular cancer. They represent about 10 to 20% of all cancers, of all breast cancers that are diagnosed. They're very aggressive, they spread to other organs, and those organs tend to be the brain and the lungs, which are never good. They're more likely to recur than other breast cancers. They're what we call high-grade tumors, in which the cells on those tumors, they don't resemble normal cells anymore. And finding target therapies for this disease is kind of like the holy grail. We're trying to do it. And so to tell you about our approach, we're using a 3D culture or spherical approach. So I have to backtrack a little and tell you what a 2D cell culture is. So you think about those plates that I showed you at the beginning. This is what one well of them would look like, and this will be the cancer cells. And so it will look like a uniform layer at the bottom of the plate. There's limited cell-to-cell interaction when you grow the cells this way. There are no nutrient or oxygen gradients, and the extracellular matrix context is mostly here at the bottom. If you grow the cells, though, in 3D culture or as spheroids or as little tumors, you maximize cell-to-cell interaction. Now you have nutrient and oxygen gradients present within this little tumor, and there's more extracellular matrix interaction. This more closely mimics tumors, and it's thought that it is going to be more clinically predictive, anything that is found in a 3D cell culture. So you can see here how our 3D spheroids look. And so if they look blue, they're alive. If they are starting to undergo apoptosis, they cleave a molecule called caspase-3 and it turns green. And if they're dead, their membrane becomes porous and they become red. So blue alive, green, they're undergoing apoptosis, red, they're dead. And so you can see that this is our control, and even in our control, in that center, there's hypoxia. There's a little bit of cell death going on because those cells are not as happy as the cells in the outside. But hopefully you will agree with me that when we treat with this compound called HB395, we're inducing a lot of apoptosis in this spheroid. So we're basically killing that little tumor with this compound. So that got us very excited. But this got us even more excited. And I know you're going, okay, that girl is really weird. Why would she get excited about that? Well, because if you look at this, so we now use increasing doses of HB395, and the orange here follows the caspase-3, so that's the green, the number of cells turning green, the number of cells turning red, and these are the number of blue cells, how many of them we killed, the decrease in their number. And this is when we do it as a spheroid. However, if we do it as in a normal way, in the 2D, and we increase the incubation now to 72 hours, you can see that I'm killing no cells whatsoever. So this compound is very selective, and it only induces apoptosis in those little tumors, but it doesn't do it to cells that are growing in the 2D. So going back to that selectivity, this is something that we want to see. So we were super excited about this compound, and we decided to do more studies to see how it was working. And for this, we use a reverse phase protein array, or RPPA. I know, big names. What we do is we get our cells treated with either a compound or the controls. We extract protein from them, and we put them into this membrane that has many different antibodies. The one that we use has about 450 different antibodies that cover different signal transduction pathways. And they have some sort of reporter that allows us to see what is being bound. So then we can compare the control to the cells that were treated with our compound, and we call this differential proteomic expression, and that allows us to know what is happening in terms of the signaling inside the cells. So bear with me, these are going to be scary slides, but I think you guys can bear with it, okay? So what does all this mean? So all the ones red are the proteins that were downregulated, are all the ones in green are the proteins that were upregulated. So we increase the levels of expression of them. And so here we're just comparing the control treated cells from cells grown as 3D to the control treated cells of cells growing as 2D. And so this is just to show you how much we change the signaling inside the cells by growing the cells as spheroids. But of course, what we wanted to know is, how is our compound doing? So now we compare our HB395 treated cells to our control cells when they're used, when they were grown as 2D or in the flat part. And so you can see that we didn't get a lot of changes, and that's consistent with the lack of apoptosis that we saw in the 2D. But all the changes that we did see, we brought down HER2 and we brought down BRCA1, which are important molecules in breast cancer, and this is in the 2D. Of course, what we really wanted to know is what's happening in the 3D. And so when we compare the HB395 treated 3D cells to the control treated 3D cells, now we're back to seeing major changes in terms of the signaling. And even more exciting for me is that many of these changes, all these proteins that were downregulated, are targets in breast cancer, in triple negative breast cancer, meaning bringing them down is better for the patients. And some of these proteins that are upregulated, if they are upregulated, is better for the patient. So this compound is causing apoptosis in small tumors and is bringing the signaling to things that we want to see in patients with triple negative breast cancer. So we took that signaling and we put it through a database on the Broad Institute in Boston. And they basically keep tabs of different compounds that they know how they act, the changes they cause in signaling, and we could compare our signaling to theirs. And so that brought us to here. And so that allows us to make hypotheses to tell us what potentially the mechanism of action would be. And this hypothesized that the mechanism of action for HB395 is as a MEK inhibitor. We still have to prove this, but MEK inhibitors are indeed have potential to be therapeutics in triple negative breast cancer. So everything is agreeing with the way we think ought to work. We went back and looked at the signaling because we had done 450 proteins. So we have many that were part of the MEK signaling pathway. And I tried to color code them here with the colors that I'm using here. But here's MEK. And so if indeed we had an inhibitor, all these proteins that are here that are what we call downstream of MEK should be in this area, should be downregulated and should be red. And you can see that this is not the case. We have some like RSK and some of the other molecules here that are being upregulated. To make it more interesting, one of the most downregulated was this DUS4, which is right here. And this is a negative regulator of ERK. So that means that if I bring this down, I'm actually activating ERK. So we might not have the right hypothesis for the mechanism of action, but we're getting insights. And so far, everything that we've seen from this compound is so interesting because it really targets triple negative breast cancer cells when they're grown as little tumors. So to bring you a summary of what we had seen with this 3D spheroid cytotoxicity assay, this type of screening is promising. We're identifying compounds that are only active against triple negative breast cancer when they're growing as tumors and not abnormally. We have a current active grant from the Florida Department of Health, the Banhead Collie program, that has allowed us to screen about 3,000 fractions from the PIC library and of course our pure compound library. And our chemists are doing fantastic progress on figuring out the compounds that are responsible for the activity. We have identified HB395 and another compound called HB157 that both only induce apoptosis when the cells are grown in 3D, but not in 2D. And this is a new activity for these two known compounds, and HB157 is more potent than HB395. So we're super excited about that. Both of them require more work, but again, very, very promising. So just to summarize everything that I covered today, you can see that through our drug discovery program, we are focusing on pancreatic cancer, which is the fourth leading of cancer death in the US through immunotherapy, B7-H4 and FOXP3. We are finding marine natural products that can target triple negative breast cancer, which is the second leading cause of cancer death among US women. And this is through the spheroid cytotoxicity assay. And you can see that we are focusing on lung cancer, which is the leading cause of cancer death in the US through identifying survivin inhibitors. So I gave you a ton of science. I know I throw you completely complicated things like arrays and signal transduction. So the next slide is the slide that you need to remember. If you know that slide, you did good, okay? And so hopefully you will agree with me after this talk that marine natural products really are promising compounds, and all these lovely organisms that I'm showing you these pictures here contain marine natural products that might help us fight cancer. So just to conclude, I always like to answer a couple of questions that I always get asked. And so the first question that I always get asked is, what can I do to avoid cancer? Unfortunately, I don't have a magic recipe. We can do very little about our genetics. We haven't figured out, otherwise I will be taller. But there are some measures that we can take to reduce our risk of developing cancer. And that is eating a balanced diet, a little bit of everything, don't exclude anything. You need some oils to absorb certain proteins. So really, a little bit of everything is the best approach. Exercising regularly activates many signaling protecting pathways that you want to have active. Maintaining a healthy weight, avoiding excessive sun exposure. You need some to produce melatonin and other proteins, but don't overdo it. Not smoking, and getting regular checkups. The other question that I get, hopefully I got you all pumped out. You want to come and be our next volunteer and say, how do I help you in your research? Well, there are many ways in you can support our research. The first and the most important is keeping our oceans and our estuaries clean. I show you some amazing marine natural products that we have identified. Think of the many more that we haven't yet discovered, and we don't want to destroy them we have before we get the chance to discover them. So let's keep our oceans and estuaries as clean as possible. You can also be a research advocate and tell others about our research. We appreciate you spreading the voice about what we do. And we also ask you, if you hear that the NIH funding is going to be cut, that you write your congressman or your representative and you ask them to maintain it. We support our research through research grants. They pay our salary, they allow us to do our experiments. So we want those to continue, and we need to have more funding. So we're always in pursuit of funding. And you can support state legislature that funds the Florida Biomedical Program. As you can see, some of that is supporting my program and is doing fantastic work, at least we think it is. Or you can make a donation if you are so inclined at the Harbor Branch website by designating your donation for the Harbor Branch Biomedical Marine Research in the pull-down menu. And a lot of people think that those donations don't matter. I can tell you, we have a local foundation called the Bernard A. Egan Foundation that has been supporting my program. through the past years. And it is thanks to them that I'm able to do experiments to pursue new directions that allow me to secure those grants that are now funding larger experiments. So with that, I just want to thank all our funding sources, including the NIH, the Florida Department of Health, the Harbor Branch Oceanographic Institute Foundation, the Cooperative Institute. We got a couple of pilot study programs, grants from the Florida Save Our Seas, the plates, and of course the Bernard A. Egan Foundation. And as I described before, we're truly a multidisciplinary group, we're a small group. So all of these people that make BMR are responsible in some way. They contributed to the research that you're seeing here. And with that, I'll take any questions.
Okay, so thanks, Esther. I think one of the hardest things for us as speakers here is we don't get to hear the response of the audience during the talk, but especially the applause. So I think, you've done this enough, what a good feeling is. So thanks so much. So for those of you, there's a reminder that you can see on your screen that if you have any questions, you can just type them in. The Q&A box is usually down toward the bottom of the screen, just off of where you can see the PowerPoint presentation. And usually, we'll go through these from where they come in. Sometimes I can find them. That makes it a little bit easier. And we've got a little bit of time for some questions. We'll try to get as many as we can. First one that came in. So early on, Esther, you said that you gave an example of something that took 46 years from extraction to the clinic. And the question is, what can be done to shorten the cycle, but also the person put it in context of COVID. We've seen a very rapid vaccine development. I mean, it's unbelievable. In response to COVID-19, but this needs to be more broadly in place.
I agree. I mean, we also have to thank our lucky stars because in terms of COVID, they were not starting from scratch. They had already done some research with other strains of SARS viruses, and they were able to use that to form the basis of the vaccines that you can see. But that being said, there was an insane amount of effort, of money that was placed towards discovering the vaccine, and that helped develop it much faster. So in case of, yes, I gave an example that is a little extreme. Hopefully it will not take that long for future discovery, but the investment that is done, I mean, as I said, you see some of the work that I've done, we're getting little by little with small grants. If we were able to get a much larger one, we could do a lot faster and get a lot more production than we're doing. Again, some of it is luck. As I said, we got very lucky with COVID. I'm very grateful for that. I cannot wait till I get my vaccine. But it is one of those things that is not always going to happen. So with natural products, one of the major problems is the supply. We need to be able to obtain more, and we cannot go back to the sea and just harvest everything that is out there. So we need synthetic chemists that are going to work on producing the compound in the laboratory or finding other ways of producing. It can be produced by fermentation, it can be produced by biosynthesis, and if truly successful, we could get cells that are going to be producing it there. But until we have those responses, some of these compounds are super complicated. So it's not a trivial aspect to get the synthesis of it. So I hope that I address some of these things. Again, it's investment, some of it is luck, some of it is also getting the right people involved. I mean, I think one of the things that was amazing with COVID was the collaboration between many different scientists that all provided their expertise to allow this effort to happen faster. And so while we work with other scientists, it's not always easy to secure that if you don't have the funding to secure their expertise.
The question is, have you looked at cancers that transferred genetically? If so, is there a way to eliminate genetic influence on activating cancer cells, i.e., BRCA2 gene is linked to breast and pancreatic cancer?
So yes, I'm trying to understand what they refer with terms of cancers that transfer hereditarily. And I think in this particular case, the BRCA1 mutation is inherited and it increases your susceptibility to develop the disease. All the cancers that I talk about contain mutations in a particular gene called RAS. And so they share that in the particular thing. So we are focusing on genes that transfer their mutations in a particular gene, and that we're changing the results of having that mutation. So even though we're not focusing on BRCA1, we're focusing on a molecule that is further upstream that does happen to control or regulate many of the pathways that are taught. So we are looking at things that do get inherited. Now, there are certain ones, we don't understand why you particularly have the BRCA1 gene. It does increase susceptibility. We haven't focused specifically on that particular kind of mutation.
Mm-hmm. Here's another question. How do you determine initially what marine species to target to achieve the highest likelihood of developing a successful cancer fighter? How do you pick what you look at?
So I have to say, so anytime that you talk to scientists, we stand on the shoulders of giants. There has been a lot of research. So if you ever go on a marine expedition with either Shirley Company or with Amy Wright, who have been working here at Harbor Branch for many, many years, they know which marine species contain more chemicals than others because they have been doing this work for a long, long time. In terms of the targets that we use specifically in cell biology, it all depends again in the research done by other groups. When somebody comes and says, now we did this research and we found that surviving, for example, is overexpressed in certain cancers, that provides what we call a target. So when I read those research papers, I know now that I have a target that I can focus on and try to study. Sometimes it's advances in technology. Until we had the high content imager, we could not do the 3D culture because we had no ways of following it. So it's kind of keeping up with the technology, the science done by other groups, also having fantastic colleagues that have that knowledge that, as I said, you go in the sub, if you were ever lucky enough to go into a research station with Amy, and she would just look at a species and she can tell you based on her experience which ones contain more marine natural products than others.
You remember Nolan? Nolan Barrett, he's here.
Hi, Nolan.
He says lots of applause. First of all, he does like your Star Wars analogy, and of course, quickly followed by Bernie Sanders. And his question, which is a little different, but talks about COVID again, is how has COVID-19 pandemic affected your work?
So, in different ways. So the funding has been critical because a lot of it has been diverted from cancer to going to COVID. Absolutely understandable, but it's making it a little tougher for us to continue. But thanks to COVID and being in self-isolation, I was able to do a lot of the analysis that you saw for the protein arrays because I didn't have anybody bugging me and I could just focus on that. So it was really fantastic in terms of the bioinformatics I was able to do. We actually have also, Amy has sent some marine natural products that have activity against COVID. So we have found some marine natural products that have that. I don't know what the follow-up is of them, but that has happened. So it has affected in many different ways. I did stop our research for a little while. We're slowly getting there. And so those will be the answers.
Yeah, and I think the answer is, we've all been doing the best we can. And,
yes.
We have guidelines and we've done pretty well overall, I think. Got a question. When did you actually begin growing cells in 3D? And how did you know that you should try 3D?
Oh, so the 3D technique is one of those that it existed a long time ago. It stopped being in vogue and people stopped doing it. And then it kind of came back into being a popular thing as high content imaging became more popular. For our lab, we started about maybe 4 years or so ago. It was after we became a little bit more comfortable using our high content imager that we decided to merge into this. And we want to try to do it with more other cells. Now, there are tricks in this. Every cancer cell line has its own little characteristics of what they like and dislike in terms of growing in as 3D, but we're seeing some amazing results. So it is something that I think we are going to continue.
This is a little different, but still related to cancer and marine organisms. Do any of these marine organisms get any type of cancer?
So I don't know that we have ever studied that.
Mhm.
So it is the same thing as when you hear, other species don't get
Always hear
cancer.
sharks don't get
Don't
cancer.
get cancer. It's like how we really studied that as much as we have. So I don't know that anybody has studied whether these marine organisms get cancer. Now, for sponge, it would be insanely difficult to determine because they are aggregates of cells. So that in itself is the definition of a tumor. So I don't know how you would differentiate that into a cancer.
So this is kind of for me, so I hope I don't put you in the spot too much, but so you talk every 3 years in the series. We look forward to that. What do you think in the next 3 years? I mean, all the work you're doing, what would be the most exciting development, just from the research side? What you're on now that in 3 years you might come back and say, you know what, here's what I did. This was the most exciting thing.
I mean, right now, as I said, we have those compounds that are looking super exciting against triple negative breast cancer. I would love if they were to go somewhere, because I think that I don't know, to my opinion, I know I'm very biased, but they deserve to be in the clinic, I mean, it's something that I would like to see more focused towards that. We're doing more, I mean, hopefully in the next 3 years, we will be able to secure funding so we can test some of this in vivo and really assert whether or not they have that probability. So that for me is really important. We have other things that we are kind of wanting to target that I hope they come to fruition. Again, it's all depending on whether or not we're able to secure that funding. But I think that that's the main thing for me. If in 3 years I can see you, Dennis, and say, hey, we're bringing this closer to the clinic, that for me, it would make me so happy. It would be the best thing ever.
We hope that's what happens, right? So I think I got through most of the questions, or all the questions. There was one that I'm going to answer as well. It got responded to already to the person who wrote it, but this gets asked a lot. I get emails like every time we say we're going to have a talk. Oh, I can't see this. I want to see a recording. Or sometimes people want to see a talk again. In this case, they want to see the talk again. So we do post these on our Ocean Science Lecture series site. What I always tell people, the easiest way to do that is just if you just Google Harbor Branch and Ocean Science Lecture, it comes right up immediately. It's at the top of the page, and you can go right there and you can go through it. And right now, Linda is sending it out to everybody if you want to see the direct link. But if you just remember Harbor Branch Ocean Science Lecture Series, Google it. You can see the talks that have been given over a bunch of years. You can even go back and see Esther's talk probably from 3 years ago if you need another Esther shot before 2 more years from now. Other than that, let me check one more time. I don't see any new ones around.
Make a comment before we, as an immunologist, I feel I have to do a plug-in for vaccines. Vaccines are safe. Please, unless you have an allergy to any of the components of the vaccine, it should be safe for you to receive your vaccine. And it is really important that most of us get vaccinated whenever they're available. So just bear that in mind and wear your mask. We do this not only for ourselves, but for others. The more we do this, yes, we all want to see ourselves normally soon. But the more we wear our mask, if we all get vaccinated, that will happen sooner than if we don't follow those guidelines.
Right, and you are an official immunologist, so you know.
Yes.
So what do you think? How do you feel about people like Fauci? I mean, you must feel really good that we have some of these.
I mean, I think that it's amazing. The work that was done is beautiful. It's amazing. It's very exciting.
Okay, well, I just want to thank, on behalf of everybody in the audience, thank you again, Esther. I've got your book for 2024. And we hope to see everybody in 2 more weeks. Thank you so much.
Thank you,